So chromatography is a topic that's part of the biochemistry protein subtopic. And so chromatography is a technique, or it's really a general term that's referred to separating things based on their physical or their chemical properties. You guys are responsible for understanding two different types of chromatography, their basic setup, and how they are used to separate amino acids. So this falls under the umbrella term of analyzing protein structures, but really what we're analyzing is their primary structures and which amino acids are present in a given protein. A couple different terms that we'll use throughout. The stationary phase. In any given chromatography setup, there's going to be part of it that does not move. There's also going to be a mobile phase, so there's going to be something that does move, and that's quite important because it's going to cause whatever you're trying to separate to move, hopefully to different degrees, and that's what causes them to separate out. And then usually you need to apply some sort of a locating reagent. This could be a dye. You might also use something like ultraviolet light to help locate things, but a lot of the times the things that you're separating may not have a color on their own, and so you have to add something to them so that they will show up and you can actually see where they've moved. So depending on the type of chromatography, we're going to try to separate things based on different properties, but the different features of the amino acids, mainly those R groups, are going to give them different properties that we can exploit and cause them to separate. And so a lot of the time we'll try to separate out a sample that we're testing, and to know which amino acids are in it, we might run some known standard amino acids alongside of it, so we can compare the distances that our unknown sample travels compared to some known samples. That would be one way to identify what amino acids are present in your sample. Another way is that there's a value we can calculate fairly simply called the RF value, or the retention factor, and we can compare the RF values that we get in a lab to literature values for RF values if we are running the experiment under the exact same conditions. So the first type of chromatography is called paper chromatography, and this is going to separate amino acids based on solubility or polarity. So the chromatography paper is just sort of like filter paper, sort of a porous piece of paper, and it's mainly made out of cellulose. Cellulose is basically polysaccharide or starch, and so that means that there's going to be lots and lots of OH groups in the chromatography paper. So the chromatography paper ends up being quite polar in its nature, which will be important for which types of amino acids want to stick to it versus stick to the solvent. The origin is going to be where we start the chromatography, or where we start the separation. So if it's a piece of paper, normally you would draw like a dotted line across it to remember where you started. Let me do that here. So you would maybe with pencil, not pen, because then the inks will actually start to separate out. So you generally would draw a little dotted line or a straight line with a pencil so that you can always see where exactly you started. You're also going to, at the end, draw a dotted line where the solvent makes it to, and we call that the solvent front. So it's pretty simple how it works. You basically have this piece of paper, a porous piece of paper, almost like a paper towel, and you're going to dip it or hang it into some solvent, some sort of organic solvent. And just like water would suck up a piece of paper towel, the solvent will move up through the paper as it gets soaked up into it through capillary action, and it gets attracted up through the paper. And after a while, when it's made it near to the top, you will stop it by pulling this off, and so the paper is no longer in the solvent. And so at that point, the solvent will stop moving up the paper. So that's where you mark the solvent front. It's where the solvent has soaked up to. So the origin's where you put your samples, usually as a solution, and then the solvent front is where the solvent makes it to. And it's all happening on this chromatography paper. So the solvent's going to be really key to making paper chromatography work properly. You're going to have to use different solvents of different polarities to try to separate out different amino acids. So some amino acids, as we've talked about before, can form hydrogen bonds. They're very polar, while other ones are extremely nonpolar because they only have hydrocarbons in their R groups. And then some are in between. And so to get them to separate, you're going to have to play around with the exact nature of the solvent to cause the different amino acids to attract more or less to that solvent, and so mix better or worse with it. The solvent that you're going to use, there's only one solvent not solvent, the dye that you're going to use in both electrophoresis and in paper chromatography, the two techniques you need to know, is called Ninhydrin. You do need to know it by name. I have a very dumb saying, as you would probably expect at this point. There's no hadden from Ninhydrin. That's what I say. That's how I remember it, because there's no hiding from it, because when you spray Ninhydrin on it, it binds to the amino acids and it makes them look blue, kind of purplish, so it makes them show up. Therefore, there's no hiding from it. So there's no hiding from Ninhydrin, and hopefully that can help you remember the name of that dye. A value that can be calculated, and this is what I mentioned on the previous page, and this is something you could compare to an actual literature value, is called the retention factor. It's a very simple calculation. With a ruler, you measure how far the spot has traveled, so from the origin to the center of the spot. So in this case, it looks like this spot has moved 4 centimeters. And then you compare that to how far did the solvent make it. So if this is the origin and this is the solvent front, and that's 10 centimeters. So the RF value for this amino acid, this little spot here, would be 0.4. It's almost like a percentage. How far did the amino acid make it compared to the solvent front? And so if you use the exact same solvent and paper and temperature, pH, all these different conditions, then your different amino acids should always move the exact same distance relative to the solvent front. And that's why you can actually have literature values for it. But mostly, this chromatography technique is fairly qualitative. You're just trying to figure out what amino acids are present in a sample, not exactly how many of each are in a given protein, just trying to identify which amino acids are in your sample. So the sample that we're testing is a protein that has been hydrolyzed, a protein that has had all of its peptide bonds chopped up and broken, usually by adding acid to the protein, and that separates out the different amino acids. And this is a picture of a chromatogram. Chromatogram is a term that can be used to describe the now separated results of a chromatography experiment. So this is a chromatogram for paper chromatography, and you can see that the amino acids have been visualized using some sort of a dye. And it's not so pretty and clean as this little cartoon version. It's usually a bit messier and looks something like this. So again, it separates things based on polarity. So things to consider is that the paper tends to be fairly polar because of those OH groups in the cellulose. And so that generally means that the more polar amino acids tend to be attracted to the paper, and therefore they don't move as much, while less polar amino acids are generally going to attract more to the solvent, and so they're going to move farther up the paper. Usually you're going to have a solvent that's somewhere in between and that tries to separate those amino acids out by subtle differences in their polarity. So gel electrophoresis works under a lot of the same principles about wanting to separate things out and looking at how far these different things are going to move, but it's not going to do it based on polarity. It's going to do it based on isoelectric points, or the charge, and a little bit having to do with their mass at given pH values. So a couple of key terms. You're going to talk about taking a gel. Okay, so the electrophoresis is not run on a piece of paper. It's run on this special kind of gel that's made, which is sort of like a porous substance. So there's kind of holes and channels throughout it, so that the given amino acids can move through it. But because of those holes, the size of the amino acid is going to matter. So small ones will be able to move through it more quickly than large ones. So that's why I mentioned mass up here. The way that you run the gel is by applying a potential difference. It really just means that you apply a positive charge to one end and a negative charge to the other end. But the technical way to say that is that you apply a voltage or a potential difference. Do not say you're going to run a current through it. For whatever reason, the IB is very opposed to that language. So this just means that you have a positive and negative charge at either end of the gel. You always have to run these with a particular buffer. So it's kind of soaked in a buffer solution. Remember that the exact pH value at which you have an amino acid will make it overall be either positively charged, negatively charged, or the overall neutral Zwitter ion form. So the buffer is going to determine which amino acids are neutral, positive, or negative. And again, we're going to use the same Ninhydrin dye to help visualize the different amino acids as they migrate. And so this is a diagram. It shows the setup of the gel, and you've got initial mixture of amino acids placed here, but then based on the exact pH, the pH being 5.7, it's going to cause different forms in our amino acids. So if we look at the 5 amino acids that are in the sample, the one that has an isoelectric pH of 5.7 is serine, and so we would expect that serine would not move, and it would stay right in this middle section where it began. Because when we apply the charge, if something has no charge, it's not going to be attracted one way or the other. We would expect that since 5.7 is higher than both of these values, so for these things, this is for these amino acids, the environment is a little bit basic. And basic, we should think of OH minus ions. We should think of that these amino acids are going to be negatively charged. And if they're negatively charged, then they should move towards the positive electrode. And we can see that that's what's happened in the diagram above. Both the phenylalanine and the glutamic acid have been migrating towards the positive electrode. Just how far they make it, it will depend on their exact charge, whether they just have 1 minus or 2 minus as their charge, and also on their mass. And these ones here, since the pH 5.7 is lower than their isoelectric points, that means that for them this is an acidic environment, which is going to cause them to become protonated and give them an overall positive charge. So they're going to move towards the negative electrode. And again, how far they move is going to depend on their mass and their exact charge, whether it's 1 plus or 2 plus. Okay. So this is sort of what I just went over. At the isoelectric pH, remember that amino acids are these Zwitter ions that overall have 0 charge because they have both positive and negative groups. If the pH is below the isoelectric point of a given amino acid, it's more acidic, and so it will be positive, and it will then migrate towards the negative electrode of the gel. And if the pH is above the isoelectric pH, the amino acid will have a negative charge and will therefore migrate towards the positive electrode. And depending on the type of charge, number of charges, and the mass, it will migrate in different directions and at different speeds. Small amino acids tend to move faster, especially the higher their charge, and big ones tend to move slower. No charge won't have any movement. Okay. And those are the different chromatography topics that you have to cover, both for SL and HL. Paper chromatography and electrophoresis, know a bit about the specifics of how they are set up and run, and be able to predict how different amino acids will move based on the given conditions. That's especially true for electrophoresis, as you should be able to predict the exact structure of an amino acid and its charge, and so figure out which direction it's going to move at a given pH.